Hi everyone. In this video, we will discuss alcohol-induced dose dumping, that is AIDD studies, why and how. Some extended release formulations can release drug too rapidly when exposed to high alcohol levels. This can cause toxic spikes in plasma concentration. Because of this risk, regulatory agencies require specific AIDD testing, especially for abuse-prone or narrow therapeutic index drugs. The goal is simple: patient safety. First, real-world cases like Peladone withdrawal triggered strict regulatory guidance. They showed how dose dumping can defeat the entire controlled release design. Dose dumping means unintended rapid drug release from an ER formulation. It happens when polymer matrix or coating fails. This can cause toxic plasma levels and serious adverse events. And in some products, alcohol is the trigger. That's why AIDD testing is now mandatory. Alcohol can weaken extended release system through multiple mechanisms. It acts as solvent for hydrophobic polymers. It reduces matrix viscosity, speeding up erosion. Alcohol can disrupt gel layers in hydrophobic matrices. Some coatings become brittle or more permeable in alcohol-rich conditions. It may also increase drug solubility, further accelerating release. Together, these effects can trigger dose dumping. FDA guidance clearly requires alcohol-induced dose dumping testing for all extended release products. Comparative profiles must prove no accelerated release in alcohol-containing media. This is especially critical for opioid, CNS drugs, and narrow therapeutic index compounds. Studies must include multiple alcohol strengths, and results must be submitted in NDA and ANDA fillings. It's a regulatory must for patient safety. Even moderate alcohol levels can weaken ER coatings, so we test a range of concentrations to ensure safety margins: 5% beer or wine, 20% fortified wines, 40% spirits, and sometimes 60% for worst case scenarios. Each profile is compared to a 0% alcohol control. This confirms no risk of accelerated release. For AIDD testing, keep the apparatus identical to your QC method. Only change the media composition. Use hydroalcoholic media. USP apparatus 2 pedal is most common. If tablets float, switch to USB 1. Closely monitor alcohol evaporation. It's faster than water, and always maintain consistent deaeration and 37-degree temperature. Controlled setup means reliable safety data. Dose dumping usually occurs in first 1 hour, so early sampling is critical. Repeatability and discrimination are essential. Compare profile in 0% alcohol, 5% alcohol, 20%, and 40% media. Evaluate time of 20, 50, 80% release. Overall, release kinetics and burst release in the first 1 or 2 hours. Use the same sampling points as QC or adjust if release is faster. Early data protects patient safety. In alcohol-rich media, hydrophilic matrices may collapse or erode faster. Enteric and SR coating can fail prematurely, so always observe tablets swelling, cracking, and erosion. Document any floating behavior, check carefully for coating rupture, and photograph or record the test for regulatory evidence. Visual proof supports your data. Not every faster release means Dose dumping. True dose dumping shows a major increase in release rate, failure of control profile, loss of lag phase, and a large early burst. If an ER product release almost completely within 2 hours, that's a red flag. This can drive plasma level far above the therapeutic range and cause toxicity. That's why a clear differentiation is critical. To prevent dose dumping, formulations use alcohol-resistant coating. Common polymers include ethyl cellulose and selected grades of HPMC. But be careful, plasticizers like triethyl citrate can migrate an alcohol and weaken the film. So, strategies include increasing coating thickness, using dual polymer system, avoiding alcohol-sensitive plasticizers, and adjusting matrix density to slow erosion. Design matters for alcohol safety. Different ER systems have different vulnerabilities. Osmotic systems generally resist alcohol, but alcohol can still change membrane permeability. So, testing is required. Floating system. Alcohol alters density and weakens gel strength. Swellable matrices. Alcohol lowers viscosity, causing faster erosion. So, each mechanism must be individually evaluated for AIDD risk. For ANDA and NDA submissions, regulators expect full transparency, provide comparative profiles with statistical analysis, clearly justify the alcohol concentrations used, discuss safety margins, and final risk conclusions, include visual evidence, photos, and videos, and highlight the formulation controls you have implemented. Transparency builds regulatory confidence. Every extended release product must prove alcohol resistance. Alcohol can disrupt polymers, coatings, and gel structure. So, we test from 0 to 40% alcohol and early worst release is a major red flag. That's why alcohol sensitivity must be considered early in formulation design. Smart formulation plus robust testing is how we prevent dose dumping and protect patients. Thank you for watching this video. With this video, we are just ending up section for dissolution method development of oral solids, and in next section, we will just take dissolution method development for complex dosage form. Please subscribe us for getting update on upcoming industry application oriented video, and stay tuned because in this 62 video series, we will cover dissolution method development, validation, troubleshooting, and many real industry examples. Please share your queries in comment section. See you in next video. Thanks.